Welcome to part 2 of histology images, where I'll be discussing about the most important histology slides for NEET PG, INICT and FMGE. So first thing is about lymph node. Lymph node, it's very easy to identify. You'll be having cortex, medulla, and in between cortex and medulla there is Okay, so this cortex is made up of the central part of the cortex is germinal center, which is paleo when compared to surrounding tissue. This germinal center is developed. It develops whenever our body is exposed to antigens. So it consists of mature B lymphocytes, right? And between the cortex and the medulla, you'll have paracortex. In this paracortex, you'll find a mixture of both immature and mature T cells. So there is immature plus mature T cells, whereas in the cortex you'll find mature T cells. Okay, this is about lymph node. Then comes spleen. It's very easy to identify spleen because if you see islands of blue in the vast red zone, then it is spleen. Okay, and this red zone is known as red pulp, which is having open type of circulation, and this bluish thing which you are seeing is lymphatic nodule, also known as white pulp. And sometimes within the lymphatic nodules you'll be able to see the splenic arteriole, right? And these are the parts of the lymphatic nodules. Okay, so the central part is germinal center. Surrounding that you will have mantle zone, and between the lymphatic nodule and red pulp, the zone which is there is known as marginal zone. So this is marginal zone, right? Okay, next one is about tonsils. So tonsils, how to differentiate it from thymus is because these two are very confusing. If you see tonsillar clefts embedded or surrounding the lymphatic nodules, then it is tonsil, whereas in thymus you don't see the tonsillar clefts. Okay, and other important features of tonsil is there are lymphatic nodules, there are tonsillar crypts, and it is lined by stratified squamous epithelium because tonsil is a part of oropharynx. Since oropharynx is lined by stratified squamous epithelium, the tonsil is also lined by stratified squamous epithelium. Then it's thymus. Then comes thymus. So thymus, it doesn't have any lymphatic nodules. Like instead, it has a lymphatic globule. Now, what is this lymphatic globule? If I'll just zoom it in, this whole thing is lymphatic globule, where you're having an incomplete septae, and the outer part is cortex, and the inner lighter or paler part is medulla. So I'll show it here. So this whole thing is lymphatic globule. You have incomplete septae, and this dark thing is cortex, and inner paler part is medulla. And the most striking feature of thymus is the presence of Hazel's corpuscle. These reddish colored thing, which if you find any reddish substance within the lymphatic tissue, then it is mostly a thymus. This is adult thymus because in adults the thymus is replaced by fat tissue. So this is about thymus. Then comes thyroid follicle. I think it's very easy to identify thyroid follicle, where you'll find a pinkish colloid. There are two types of thyroid follicle, right? Active and inactive. How to identify it? Like, you cannot identify, but you should know that in active thyroid follicle is having cuboidal lining, and the colloid is scalloped because the colloid is being used up to make thyroid hormone, right? So if you find scalloped colloid, it's mostly active thyroid follicle. Inactive thyroid follicle, it has got a squamous lining, and the colloid is not scalloped, right? And this is about parathyroid gland. So parathyroid gland is having two types of cells: oxyphil cells and chief cells. Oxyphil cells are the ones which are having a very pinkish material in them, that is eosinophilic granular cytoplasm with a dense nuclei. These are oxyphil cells. Then the paler cells which you are seeing here is, these are known as chief cells, which are chief cells are the ones which produce parathyroid hormone, right? Then comes pancreas. So the pancreas, this is actually a previous INI-CET question. So if you see pale tissue or pale tissue in the vastness of dark, that is basically, if you find islands of pale cells surrounding the dark cells, then it is pancreas. So I'll just zoom it in. So these are these are basically islets of Langerhans. These are islets of Langerhans, right? So that is how you can identify pancreas. So these islets of Langerhans, they are mostly found in the tail part of the pancreas, right? Okay. And islets of Langerhans, again, there is a division of cells within the islets of Langerhans. The central part is made up of B cells. So way to remember is just remember B in center. B in center means B cells are found in the center, and in the periphery the alpha cells are found. Like alpha males are guarding the territory in the periphery. You can remember in that manner. In the periphery, alpha cells are found which will secrete glucagon. Other cells are PP cells, which are pancreatic polypeptide, and delta cells, which secrete somatostatin. So this of the tongue. So tongue is lined by stratified squamous epithelium, just like tonsil, because it is a part of oropharynx, right? But the dorsal surface of the tongue has got different types of papillae. So broadly, there are 4 types of papillae which are found. The first one is filiform papillae, which has got a conical projection. They are small, and this is filiform papillae, where you can see there are many conical projections found. And important point about filiform papillae is it doesn't have any taste buds. Because way to remember this is filiform, it sounds like it is already filled with lots of taste, so it doesn't have any taste for any space for taste buds now. So filiform doesn't have any taste buds. Then comes fungiform papillae. Fungiform papillae, the taste buds are found on the surface. So you can remember it in this manner, like fungus usually grows on the surface of the food, right? So in fungiform papillae, the taste buds are found on the surface. Okay. Then comes a foliate papillae. Foliate papillae, the taste buds are found in the grooves. So this is an example of foliate papillae where the taste buds, you can see here taste buds, right? So they are found in the groove. So this is an example of foliate papillae. Now, then there is a last one is circumvallate papillae, which is the largest kind of papillae, which is mushroom shaped. It's mushroom shaped. And another way to remember is when you extend it down, it kind of has this conical projection downwards. Okay. And again, the taste buds are found on the sideways. It is the largest papillae, and this is found anterior to sulcus terminalis of the tongue. Okay. So important point about filiform is it doesn't have any taste bud. Conical projection. Fungiform, taste buds are found on the surface. Foliate, they are found in the grooves. And circumvallate, mushroom shaped, is having largest it is the largest papillae. Then comes the esophagus. Esophagus is lined by stratified squamous epithelium. Okay, and then next part is lamina propria. Under that you have muscularis mucosa. Then this whole part is submucosa, which has got glands in it. So these are the glands which are found in the submucosa. Then this whole part is muscularis propria, where the inner part is lined by circular muscles, then the outer part is lined by it consists of longitudinal muscles. These two are the images of duodenum, whereas you have long villi and crypts in the duodenum. So these are the villi and crypts in the duodenum. Most importantly, in the duodenum you will find these glands which are known as Brenner's glands, which are found in the submucosa. This is about duodenum. Then comes jejunum. Jejunum has got this peculiar fold which is known as plica circularis. This is nothing but the fold of submucosa and the mucosa that are protruding out, right? And rest of the things are or layers are seen. You have submucosa, muscularis propria, again which is lined by inner part is circular muscles and outer part is longitudinal muscles, right? And one more thing which you should note is in the submucosa you are having Meissner's plexus, whereas in the muscularis propria you will have this Auerbach's plexus. Submucosa, way to remember it is in the SM. Remember, in the submucosa you are having Meissner's plexus, right? Then comes ileum. It's easy to identify because in the ileum we'll be having this lymphatic tissue known as Peyer's patches found in the antimesenteric border of the ileum. The rest of the layers are the same. And one more thing about the GIT lining is in the intestine you have this Paneth cells. I'll just zoom it in. these are like pinkish granules which you are seeing here. They are actually defensins and lysozymes found in the Paneth cell. So eosinophilic material in the mucosal crypts containing defensins and lysozymes, these are nothing but as Paneth cells. Kidney. Um, uh, how to identify the histology of kidney section is, if you find the tissue, and this tissue, if it is surrounded by all the dilated tubules, again in this section you see some tissues surrounded by all the tubules, right? Then it is kidney, a section of kidney. And this tissue which you are seeing here is glomerulus. So glomerulus is basically tufted capillaries, which contains, again here contains tufted capillaries, and the space which is which you see here surrounding the glomerulus is Bowman's space, right? And surrounding this you see PCT and ACT. It's not asked in exams, but it's good to know how to differentiate between PCT and DCT. PCT is having a hazy appearance because of the presence of microvilli and or brush border, whereas DCT is having clear appearance. I'll just show it in this picture. So this is PCT, which is having a hazy appearance, whereas this thing is DCT. Again, I'll show it here. This is PCT, this is PCT, PCT. This is DCT, which is having clear appearance, right? This is about kidney. Next one is about the liver, microarchitecture of the liver. So this liver is having the portal triad and the central vein, right? This portal triad is made up of bile duct, portal vein, and hepatic arteriole. And in between the sinusoid and hepatic cells, there is a space that is known as space of Disse, right? Space of Disse is it contains ito cells, right? Ito cells has got different functions in different stages. In dormant stage, it will just store the vitamin A, whereas in active stage, for example, if the liver is injured, it will lay down the type 3 collagen. So it will lay down type 3 collagen, whereas in dormant stage it is storing vitamin E. This is an important question, right? One more thing about liver is it has got Kupffer cells, which are nothing but as macrophages of liver. They are known as Kupffer cells. A histology of fallopian tube. It's very easy to identify. If you find these mucosal folds, if you see this mucosal fold surrounded by muscle tissue, then it is fallopian tube. So fallopian tube has got three layers, right? Mesosalpinx, also known as serosa. Then you have myosalpinx. The name itself suggests that it has muscular layer. Inner is circular and outer is again longitudinal. Endosalpinx is made up of all the mucosal layer, which is having numerous branches or foldings like this, right? And it has got a ciliated columnar epithelium. Also peg cells, which are non-ciliated epithelium, which help in capacitation of the sperms, right? This is about fallopian tube. So just remember the foldings present in the fallopian tube. Then comes seminiferous tubules. Seminiferous tubules, basically within or in between the tubules you find the interstitial space, which has these cells known as Leydig cells. And Leydig cells has got these pinkish material known as Rinke's crystal. Okay, Rinke's crystals are found in Leydig cell. This is an important question. Then within the seminiferous tubule you will find there is a spermatogonium. Then it will undergo mitotic division. Remember, it is mitotic division. It undergoes mitotic division to form primary spermatocyte. Then it undergoes meiotic division to form secondary spermatocyte. Then again it undergoes second meiotic division to form spermatid. Ultimately it matures to form spermatozoa. In the tubule you also find Sertoli cells, which are nursing cells, or which will nourish the spermatocytes, right? Adenoids are a part of nasopharynx, right? Since it is a part of nasopharynx, it is lined by pseudostratified ciliated columnar epithelium, unlike tonsil, which is lined by stratified squamous epithelium. Okay, the adenoid, since it is a part of nasopharynx, it is lined by pseudostratified ciliated columnar epithelium. In the adenoids, you can find lymphatic nodules. These are the lymphatic nodules. These are all nodules. And sometimes with adenoids, you can also see the presence of the glands. These are the glands of the pharynx. Remember, it is not tonsil. Why? Because the lining is not stratified squamous epithelium, and it doesn't have any clefts, tonsillar cleft. It is not thymus. Why is it not thymus? Because it is not having that thymic lobule that I had explained in the earlier part of the video, right? So if you find lymphatic tissue along with some glands, it is most likely to be adenoids. See, these are the histological images of endometrial phases. The first one is early proliferative phase, where you see glands which are in the form of a tube. So these are known as tubular glands. Okay, this is seen in early proliferative phase. Then comes early secretory phase, wherein you can see subnucleolar vacuolation, and the glands will also become a bit dilated. So this subnucleolar vacuolation is the first sign of the ovulation, which you can see in endometrial biopsy. So this this vacuolation which you are seeing under the nucleus is known as subnuclear vacuolation. The third stage is late secretory phase, where the glands will appear in the form of corkscrew. So these are known as corkscrew glands. So these are all corkscrew appearance of the glands seen in late secretory phase. This is a histological section of adrenal cortex. There is a famous mnemonic from outwards to inside, GFR. So G stands for glomerulosa, F for fasciculator, R for reticularis, right? G is the one, zona glomerulosa will secrete the mineralocorticoid hormone like aldosterone, which is stimulated by, this is an important question, aldosterone hormone production is stimulated by hyponatremia, hyperkalemia and angiotensin 2. The second layer is fasciculata, zona fasciculata, which will secrete the cortisols, basically glucocorticoid hormones like cortisol, and it has got intracellular lipid droplets because the cortisol is synthesized from the cholesterol, right? So that is the reason why it has got storage of lipid droplets within the cells, and this is under the control of pituitary gland, whereas this is not under the control of pituitary gland. This zona glomerulosa is not under the control of pituitary gland, whereas this is under the control of pituitary gland. The secretion of this is under the control of pituitary gland. Then comes zona reticulata, which will secrete DHE, DHEAS. This is just the histological section showing Meissner's corpuscle in the dermis, right? And this is Pacinian corpuscle, which is in the form of an onion ring. And types of glands. In part 1 video, I have told about parotid gland is an important example for serous serous acini. Basically, serous gland example for serous gland is parotid, and mucinous gland example is sublingual, and mixed glands example for mixed gland is submandibular and sublingual. This is an important question, and these glands, especially the submandibular gland, is having serous stimulons. Now what is the serous stimulons is these are half moon shaped cellular formation in the salivary glands. So they are made up of serous cells which are attached to the mucous acini. So this is mucous acini, surrounding that there is serous acini also. So this thing is known as serous demilume. That is demilume in the sense it is half moon shaped. So I'll show you another example. Okay, this is mucous acini. Surrounding that you have serous acini. So this is known as serous demilume. So mucous acini, surrounding that there is serous demilume. So this is maximally found in submandibular gland. Also it is found in sublingual, but maximally this found in submandibular gland. Now we will speak about the types of exocrine glands. Right, there are 3 types of exocrine glands. The first one is merocrine gland. Merocrine gland is also known as eccrine or epicrine glands. So the secretions are released by a process known as exocytosis from the secretory vesicle. So the example for merocrine secretion includes one thing, a mnemonic to remember is merocrine sounds like pro. Mero is like pro. Mero pro. These are like rhyming words like mero pro. So the milk protein casein is secreted by this mechanism, or it is an example for merocrine gland. So secretion of milk protein by that is casein by mammary glands is an example for merocrine gland. The other ones include the sweat glands, salivary glands, and exocrine pancreas. Second one is holocrine gland, where holo means whole. So whole of the cell will disintegrate. That is how the secretion is released. This is the holocrine secretion. So example for this is sebaceous gland. Sebaceous gland is an example for holocrine secretion or holocrine gland. Then comes apocrine glands. Apocrine gland, the name itself suggests apo means the apical parts of the cells are shed off to discharge the secretion. The apex will be shed off to help in secretion. Apex, okay, and apo sounds like lipo. So the secretion of milk fat by the mammary glands is an example for apocrine glands. Also, another way to remember is the apex of a body is having hair. So sweat glands in the hairy region like axilla and inogenital regions, they are also an example for apocrine glands, whereas ecrine sweat glands is an example for merocrine gland, whereas the sweat glands found in the hairy region of the body like axilla inogenital region is an example for apocrine gland.